Hello everyone and welcome to a second part of Area Study 1, which is looking at how we can manipulate all of these processes that we've learnt about for medical and scientific purposes. So this is on chapter 4. So I've split chapter 4 into 5 separate videos for you. So DNA is a marvelous set of genetic instructions that we use to produce proteins. We've gone through this in chapter 2. Over the years, our understanding of these processes like DNA and proteins and enzymes and how they all function has skyrocketed so much that we've been able to start using these processes to manipulate things in science and medical endeavours. We know how these molecules function in the body, so how can we use these functions to help us do incredible things in the lab? So over the next couple of videos, we're going to be looking at specific techniques that we have observed in humans or particularly bacteria, and we're now manipulating those processes to help us scientifically. So the first thing that we need to be able to do is we need to be able to define what DNA manipulation is. So it's sometimes also referred to as genetic engineering, and it involves any direct manipulation of an organism's genome using biotechnology. So we can introduce genes, we can edit genes, we can remove genes, and basically the main aim of doing this normally is to enhance an organism or to improve an organism or to introduce desirable traits. So if you look on the right hand side here, this thing at the top, which I personally wouldn't recognize if I saw it, is corn. It's corn as it naturally grows in the environment. And over the last couple of decades, we have genetically modified corn to what it is today. And we've done that by introducing a range of genes that help it to grow and help the corn be like bigger and juicier, more nutritious. So that's an example of a genetically modified organism. So we often shorten it to GMO. So even things like corn, which we eat every day and don't even think about, are genetically modified organisms, and their DNA has been altered over time. So the first dot point, the first technique that we're going to be looking at is the use of enzymes to manipulate DNA. So we're going to look at 3 different types of enzymes. But before we do that, I'm going to give you a quick enzyme rundown because it might have been some time since you've looked at enzymes, and we're going to go through enzymes in a lot more detail in chapter 3, but I'll give you a quick rundown so you know what we're talking about. So enzymes are proteins. Okay, we know lots about proteins. We know that their shape is really important and their very specific function. Enzymes are proteins that act as biological catalysts by accelerating chemical reactions. So enzymes aren't used in a chemical reaction, they're just there to facilitate and accelerate it. So there's 2 different examples of what enzymes can do here. So enzymes can catalyze anabolic reactions. So anabolic means to basically to build something up. So you'll see in this diagram here, we have 2 separate substrates. So that's what, so anything that is, catalyzed by an enzyme is called a substrate. We have 2 different substrates that are going to bind to this enzyme, and then you can see that they have created a product. So 2 separate substrates have been built up to create this product. So what goes into the enzyme is called a substrate. What comes out is a product. And in an anabolic reaction, we are taking 2 substrates and merging them into one final product. We can also use enzymes for catabolic reactions, which are like the opposite. So that's when we have our substrate is a molecule of some description, and then the enzyme is going to be used to break this into 2 separate products. Okay, so it's important that you understand anabolic and catabolic because we'll be talking about those as we go. So there's 3 different types of enzymes that we can use to manipulate DNA. So these are endonucleases, ligases, and polymerases. So here I really want to note the suffix that we're using for all of these. So the end of all of these enzymes is the suffix "ase." And this is a really important thing to note because often in exams they'll ask you about an enzyme you've never heard of before that has "ase" as a suffix, and you'll need to be able to identify that that is an enzyme that they're talking about. So this is a really good note to make, that if you see "ase" at the end, we're usually talking about an enzyme. So the first enzymes we're going to be looking at are something called endonucleases, and they're often referred to as like the genetic scissors, if you will. So these enzymes are responsible for cutting DNA, just like scissors. Okay, they're sourced from bacteria. So endonucleases have originated from bacteria. We observe them and we now use them for our own purposes. But in bacteria, they were originally used as a defense mechanism against viruses. So we're going to learn about this in our next chapter, but bacteria can use these endonucleases to snip viral DNA. They also can be sometimes called restriction enzymes, and they will target specific recognition sites on DNA. So you will see in this diagram in a minute what I mean by that. So to cut the DNA, the enzymes cleave the phosphodiester bond on the sugar phosphate backbone that holds DNA nucleotides together. So I'm hoping that those words are clicking in your brain. So do you remember when we went through, and this is a very simple ladder version, we went through the structure of DNA, and we remember that they have a phosphate and they have a sugar, and then they have bases, and they're all connected. And there's another one, and then this way is going in the reverse, and then we have a phosphate, and then we have a sugar, then we have a phosphate, and then we have a sugar. So do we remember that these links, okay, or this is called the sugar phosphate backbone, so that's like the rungs of the ladder, and these are connected through phosphodiester bonds, okay? So what restriction enzymes do is they cut those bonds, which is cutting the DNA in half, essentially. So there's two different ways that a restriction enzyme can do this. They can do this with a blunt end, okay? So we're looking at this section here, all right? So you'll see that this restriction enzyme, they have funny names. You don't really need to remember any of them. But this restriction enzyme has a specific recognition site that's highlighted in orange. So every time that this restriction enzyme sees this specific sequence in a piece of DNA, it's going to cut it. And this is a blunt end cut. So this restriction enzyme is going to cut the DNA directly in the middle of its recognition sequence, and it's going to leave two blunt ends. Okay. Then we have the sticky end endonucleases. Okay, so we're looking at this example here. All right, so we've got a different recognition site here. So the recognition site for this restriction enzyme is, again, the sequence highlighted in orange, is this site here. And these produce sticky ends. So they don't cut directly in the middle. What they do is they do a staggered cut so that there is a nucleotide hanging off each side. So when you see a sticky end endonuclease, you will need to make sure the cut is leaving one nucleotide hanging on either side. And this is actually really advantageous because if we have a free nucleotides hanging here, it's a lot easier to insert genes. The blunt end cut's a little bit more tricky, but if we have free nucleotides, it's very easy for us to just slip the complementary nucleotides in, and it's way easier to insert a gene. So for our course, you need to understand how to make a blunt cut and how to make a sticky end cut. But you'll never have to remember a restriction enzyme, and you'll never have to remember the recognition site. You'll be given this information, and you'll just need to be able to make the cut. So that's what endonucleases are for. Ligases are almost like the opposite, okay? So when the endonuclease comes and snips the DNA, the ligase comes and puts it back together again. So it's like the glue of our genome. So they are enzymes that join two fragments of DNA or RNA. So you can see in this example here, this is the cut that we just made with these restriction enzymes. What ligases do is they catalyze the formation of that phosphodiester bond back again. So their job is to reattach those phosphodiester bonds in the sugar phosphate backbone and merge them together so that the DNA is back to its double-stranded form again. So the endonuclease is cut, and then the ligases join back together again. And they can join it whether it's a sticky end or a blunt end, they don't mind. Last but not least, we have our polymerases, and hopefully this word is ringing a few bells because we've looked at one before. We've looked at RNA polymerase when we were doing transcription, but there's a whole range of polymerases. So what the polymerase's main job is that it adds nucleotides, okay? It can either do that to RNA through transcription, or it can do it to DNA through DNA polymerase. So RNA polymerase, as we know, involved in transcription to produce RNA. DNA polymerase does the same thing, but its job is to replicate or amplify DNA. And we're going to learn a little bit more about why that's important later. So hopefully this helps. Please bring any questions to class and I will see you there.